So what actually is a neurotransmitter? One of the most remarkable things about neurotransmitters is that they have to be made of widely available, chemical materials, chemicals that are widely available in the brain. Because they're synthesized, stored, released, and then broken down, again and again and again and again. I mean, you got lots of synaptic activity occurring all the time, so you have to have chemicals that are actually pretty, abundant, basically, in the nervous system. So it turns out that many of these are, short proteins, or they're made of amino acids, or they practically are amino acids, or they're made of byproducts of fat metabolism or of cellular respiration. They're made out of chemicals that are widely produced and available, basically, in your brain. So these are widely available chemicals often. So how do they become a neurotransmitter? Well, there are going to be always some exceptions to this process, because there are some very unique, what are called non-classical neurotransmitters that have been identified, including the cannabinoid neurotransmitters, which are, basically, impacted. Their receptors are impacted by chemicals that are found in cannabis, right, in marijuana, that have a very unique kind of pharmacology. But neurotransmitters in general, this is what goes on. There's most typically like a synthesis process. So, whatever the widely available raw material is, it's abundant in every cell pretty much, but in the cell that's going to utilize, that chemical as a raw material for the manufacture of a neurotransmitter, you've got to have the expression of a gene or genes that produce enzymes, synthesizing enzymes that will grab onto that abundant chemical in the neuron that's going to release that specific neurotransmitter and convert it, alter it, attach it to something else, or, move parts around or something, create, synthesize, manufacture the specific neurotransmitter. So there's a synthesis step that's essential in any neuron that's going to release a particular chemical as a neurotransmitter. Wildly abundant, chemical raw materials, but in the neuron that's going to release that neurotransmitter, it's going to express specific genes that'll grab that abundant raw material and, convert it into the actual neurotransmitter itself. Now, part two is storage. Now, you just don't typically have it sort of wandering around in the cell, although, again, the cannabinoids are kind of interesting. But in general, this synthesized neurotransmitter then has to be stored. And when you think storage of neurotransmitter, I want you guys to think of vesicles. Neurotransmitters are typically stored in vesicles. And the way that the neurotransmitter gets into the vesicle, right, is through a transporter, because the vesicle is a phospholipid membrane-bound, kind of spherical organelle inside the axon terminals of these neurons. And, to pack the vesicle, to get a high concentration of the neurotransmitter inside the vesicle, you're going to require energy in the form of ATP to run a transporter, which can keep pulling that synthesized neurotransmitter inside and pack it up inside those, specific vesicles. So step one is synthesis of the neurotransmitter from some widely available, raw material, chemical material. Step two is storage, via a vesicular transporter, typically inside the vesicle in terms of high concentration. And then there is release of the neurotransmitter, and that's typically upon the arrival, ideally upon the arrival of an action potential, a depolarization, that arrives and opens those voltage-gated calcium channels, which initiates, changes in the docking complex, which bring, the vesicle membrane close to the presynaptic terminal membrane. There's fusion, right? There's suddenly a hole from the vesicle, which is packed with, neurotransmitter, to the outside, to the cleft itself, where there's no neurotransmitter or very little. So there's a concentration difference, right? And the concentration gradient force will drive that packed neurotransmitter, basically out into the cleft. And then there is almost always some mechanism or mechanisms for the removal of the neurotransmitter because chemical communication at a synapse, remember you got a presynaptic terminal, a postsynaptic terminal, you're getting the release of your neurotransmitter now that was stored in vesicles out into the cleft here. There's astrocytic membranes wrapped around this particular synaptic connection. It's supposed to be point to point and to some extent time limited. It's not supposed to be like a hormone that's just being released all the time and diffusing all over, etc. This is actually arrival of an action potential, release of neurotransmitter, stimulation, and then stop. So the stopping, the removal of the neurotransmitter can be accomplished in a number of ways. Oftentimes there are degradative enzymes. These are enzymes that'll grab the neurotransmitter and break it down so it can no longer stimulate its receptors. In other cases, there is a transporter. This is not the vesicular transporter, which is on the vesicles, right? That pack the vesicles with neurotransmitter and are part of the storage of neurotransmitter. But this is a transporter that's localized oftentimes on the presynaptic terminal membrane. Sometimes they're localized on astrocytic membranes. They can be in different places, but their goal is to pull the neurotransmitter that was just released basically out of the synaptic cleft and then back, let's say, into the presynaptic terminal. And in some cases, we're going to see when it comes to, for example, the monoamine neurotransmitters, dopamine, norepinephrine and serotonin, we're going to see there are these what are called reuptake transporters. These are the transporters that are embedded in the presynaptic terminal membrane that grab the neurotransmitter that was, released into the cleft and pull it back into the axon terminal, where it was synthesized. And then inside that presynaptic terminal, there's even a degradative enzyme. So there's another step. It can be broken down further. It can be, removed from the cleft and then actually, enzymatically broken down by a degradative enzyme. So synthesis, storage, release, and removal. Now I want you to look at this image. It's on your D2L page here, your course web page here. But basically this shows you a presynaptic axon terminal. It shows you a cleft, and it shows you a postsynaptic dendrite. And I just want to review really quickly at the presynaptic axon terminal. It mentions neurotransmitter synthesizing enzymes. That's where you're going to find a lot of them anyway. The synthesizing enzymes are expressed by genes. They're back in the nucleus, the genes, right? And they're expressed and then transcribed and then translated into these enzymes and then transported down often into the presynaptic terminal, where they can grab onto the widely available raw material at that presynaptic terminal and synthesize our neurotransmitter. Then it's a synaptic vesicle transporter, right? These are the vesicular transporters, which are localized to the vesicles membrane. They utilize ATP like all transporters do, but they utilize that chemical energy to kinetically force the synthesized neurotransmitter inside the vesicle to pack it, to fight that concentration gradient force, but to store that concentration gradient force too for later, release of neurotransmitter, during exocytosis. It also says here reuptake transporters. That's another kind of transporter, right? It's another transporter, but it's localized in a different spatial location. It's not on the vesicle. It's not a vesicular transporter that packs the neurotransmitter in the vesicle. It's on the presynaptic terminal membrane, where it can pull back, remove the neurotransmitter that's been released into the synaptic cleft. Then there are degradative enzymes, right? And these are found within the presynaptic terminal also often. We'll see other examples where they're actually found in the cleft itself. But these degradative enzymes can then break down, a neurotransmitter that's been reuptaken, and that literally is how they say it. Scientists are not necessarily English majors. So there's sometimes, a two-step removal process involving reuptake, like for serotonin or dopamine or norepinephrine, we'll see. And then, followed by degradation or some degradation in the presynaptic terminal. And then on the postsynaptic side, we see transmitter-gated ion channels, right? That's another, way of saying ligand-gated, ion channels or ligand-gated channels, right? Or another way of saying ionotropic receptors, right? These are the receptors that'll bind the released neurotransmitter and then it'll result in a change in shape that involves opening of a channel that can, within the structure of the receptor, right, that can allow ions to pass through. So this can rapidly depolarize, rapidly hyperpolarize. Then it says here G protein coupled receptors, GPCRs, right? This is another class of receptor, right? This is also called a metabotropic receptor. And there's it's got a portion here in the cleft, right, or the outer portion of the cell here, the postsynaptic cell, and then it's got a G protein, either GI or GS or GO, some kind of a G protein coupled to it on the inside. When the neurotransmitter binds, well, there's a shift in shape as well, but this releases a G protein, right? The first messenger inside that postsynaptic cell, which can then activate a second messenger, which can activate a third messenger and fourth, and can ultimately, send some of these, activated chemicals downstream in the biochemical cascade into the nucleus, right, where it can bind to DNA. It can, open, segments for expression or close other sections to, reduce expression. It can upregulate or downregulate the expression of genes and, the manufacture of proteins, including more, receptors, for example, right? It can change the physical structure and thus the function of the cell and the synapse. So it says here, G proteins, they were coupled to the metabotropic receptors, or the GPCRs, before the neurotransmitter binds, right? There are G protein gated ion channels. So much complexity, right? So basically here is a metabotropic receptor, or a GPCR, G protein coupled receptor. Here's our neurotransmitter that's been released, and has not been reuptaken or degraded. It binds here, shift in shape. Here goes the G protein, and it can move along the membrane. And I'm going to move this over here. This is another machine now. This is a G protein gated ion channel. This can bind to here and open a channel, open a hole. So there can be a depolarization, right, or a hyperpolarization. And it takes a little longer to initiate, but because these G proteins are released, and depending on the amount of activity, it may last a little longer, than just depolarization or hyperpolarization through an ionotropic receptor. And finally, this is second messenger cascades. Those are the biochemical cascades that I referred to, that are initiated, sometimes or most, very often by, the stimulation of a metabotropic receptor and the release of the G protein, right? That'll stimulate those second messenger cascades or biochemical cascades, but in some cases, actually, an ionotropic receptor, right? A ligand-gated channel that opens a hole, right? It can let in a particular ion, calcium, which is not found in high concentration inside, free floating in the neuron. That calcium can actually, if when it enters through, it'll depolarize because it's a positive ion, but it can also bind to some other chemical and sort of activate it. And then that can bind to another chemical and activate it. So that entry of calcium, through the ionotropic receptor, can also initiate a biochemical cascade or a second messenger, cascade. So lots of complexity at the synapse. What is a neurotransmitter? Well, it's something that is synthesized, stored, typically in vesicles, released upon the arrival of an action potential, and then removed by some mechanism, like a reuptake transporter or a degradative enzyme or both.